One of the brightest spirits in the world today is my friend Dr. Zach Bush. He is creating a vision of what's possible for humanity that millions around the world are getting behind, and for good reason. He has the ability to see clearly what the challenges are and then see past those challenges and see how we can get to higher ground. And then he weaves together through his words and thoughts a beautiful vision for what could be in store for us. Doesn't mean that what we're facing right now isn't challenging. It is, but if we can understand it and then have a vision for transcending it, that's our best hope, and that's what Dr. Zach Bush brings when he speaks. So I am very excited to introduce you to part one of my two-part interview with Dr. Zach Bush. Zach, thanks so much for doing this. I'm really excited to have this conversation.
It's a pleasure to be here again.
So let me just clarify up front, you don't represent yourself as a vaccine expert or a part of the quote-unquote anti-vax movement, but you have a lot of perspective about the microbiome, immunity, how we coexist with microbes, et cetera, which might inform us as we're thinking about these issues. So tell us kind of maybe starting with the big picture, what you see.
The big picture right now is that it has never been more exciting to be a scientist in history, I believe. There's so much revolutionary perspective emerging from the scientific world right now, and it's really being handed to us through genomics rather than a microscope for the first time.
What does genomics mean?
Genomics is a look at the way in which life templates itself and, more importantly, perhaps adapts. And so genomics or genetics is the study of how the blueprint for life merges and adapts with other life forms to create something of a higher state of function. And we've seen this since the origin of the most tiny, fundamental pieces of biology that would be arguably either the archaea, which are these tiny little ancient bacteria, or the viruses themselves. Mm-hmm. And in both cases, their entire purpose is to accelerate gene transfer. And so in many ways, genomics is the study of the language of genetic transfer, and it's the language of life in many ways. And interestingly, if we back up and look at where we've been over the last 150 years at least, we've had this perspective that we're at war with the microbiome. We're trying to beat back infection. We're trying to beat back viruses and bacteria and fungi and the like to protect this holy of holies, which is thought to be this sterile human experience. That has completely broken down through the study of genomics, which is so interesting again, because I think up until this point, we've really relied on the microscope to give us a deeper and deeper look into what is the fabric of life. And then we've delved past the biology into the quantum physics by crashing atoms into one another and trying to find that fabric of life. Mm-hmm. And what's starting to emerge from the study of genomics and realizing the plasticity or the fluent state of affairs in genetics, we're starting to realize that life has nothing to do with the fabric and has everything to do with community. And it is only through biodiversity that we see the emergence of life as we understand it on the planet. And the language of that biodiversity is genomics, and it's expressed through the microbiome and the virome in its highest state.
What's a virome?
The virome is an important description of the pool of genetic information that travels around the world in an airborne state and then absorbs into biologic systems. And so the virome is a very important differentiator from the microbiome. For too long, we've been clumping, as scientists and physicians and public, the viruses into this concept of bacteria and fungi and infectious agents. And so we have them as piece of the microbiome. People would tell you that that's bacteria, fungi, yeast, parasites, and viruses, or that kind of list. And the very definitions of the words micro and biome demonstrate that we can't include the viruses therein, because the first word meaning small, the second word meaning living organism. Biome meaning a living organism, and it turns out the viruses are not alive. And the way in which we can define that is they can't reproduce and they can't produce energy, they can't consume any source of fuel per se or the rest. And so they're a simple genetic packet of information that's transferred through biology to another species, or most often to reinforce a genetic shift within a species. And so we package these information genomic messages in smart targets. And so we will cover it in proteins that will bind to very specific receptors in very specific tissues in very specific organisms outside of ourselves. And so in recent months, we've seen the most extraordinary global attention that's ever come to the virome. We've created this story about a pandemic that was going to attack humanity, and we've created the most extraordinary economic shift in the history of mankind. Trillions and trillions of dollars have shifted hands in a very short period of time, all because of a story around a tiny little virus that is a tiny bit of genetic information that actually has no ability in and of itself to change the course of human history. There's no way that we're going to see this virus change the rate of population growth on the planet, for example. It's not going to be like the plague, where we saw 15 to 20% of all Western countries collapse in their populations. This virus from its outset was going to put a challenge on the human experience this year, but it was never going to threaten the fabric of society because the genetic information was targeted to a very specific part of biology. And so it's exciting to know that the virome is this pathway of information that is highly specific in its delivery mechanism. It's not just like this exudate from biology of stress that then is just attacking everything. It has to be absorbed through a very specific receptor, the ACE2 receptor in the case of coronavirus, and that happens to be expressed some in the lung, but also in the vascular tree and other places of the body. And so there's this eloquent information stream of genomics being updated into very specific parts. It's just like when you get a software update on your computer. It's not going to go and put that piece of code in every area of your hard drive. It's going to go and put that in a very specific place. And so around that piece of code is a targeting device that the programmer will build to push that to the right place so that it ends up at the right update. And viruses look to be designed exactly like that. These are intelligently designed, very meticulously designed updates to very specific parts of biology.
It's interesting because I was picturing, I said, wow, the virome sounds like Wi-Fi for biological upgrades, throughout the planet. But we have taken this very militant view. In other words, we have to eradicate, in this case, coronavirus or polio, or name a virus. We're at war with it. I mean, literally, we use military language when it comes to dealing with this. And based on what you're saying, is that completely just the wrong paradigm? I mean, should we really have a different look at this?
This is one of the reasons it's so exciting to be a scientist right now. The only time in history that I can look back from my scientific worldview to see a similar seismic shift in perspective was Pythagoras 2,000 years ago with the ancient Greeks, who suddenly realized that the geometry of a flat planet wasn't working. And he suddenly realized that this planet might be round, And this was then 1,600 years before we would really start to understand the mechanics and the possibility of a round planet. And that would become the discovery of the telescope, obviously, in the early 1600s. And suddenly we have these scientists who are looking out into the cosmos to realize that we weren't the center of the universe. And so it's only been 400 years since we discovered that the planet Earth wasn't the center of the universe and everything was cycling around us. And I believe we're in a very similar moment right now around the understanding of human biology on Earth, suddenly realizing we're not at the center. And we are now realizing we're a tiny planet in the vast solar system, vast universe, galaxies, etc., of biology on this planet. And so we are this tiny pixel of life, and nowhere more is that obvious than when we start to really study the virome. And the virome is showing us just the sheer amount of genomic information on the planet at any given moment. Current estimates are that we have 10 to the 31 viruses in the air around us. 10 to the 31.
10 to the 31 is that's an inconceivable, 31 zeros.
31 zeros after a 1.
Can you give context for that?
Yeah. The only number that maybe can start to put it in perspective for me is that 10 to the 31 is 10 million times more than our stars in the entire universe.
The entire known universe.
Known universe. So you've got billions of stars within every single galaxy, billions of galaxies. Multiply all of that out, you start to get a sense of a vast number. And then you have to put, another series of zeros. You have to multiply by 10 million times before you start to reach just the viruses in the air.
Wow.
And then stunningly, it turns out that we have to come to terms with 10 to the 31 viruses in ocean water and 10 to the 30 viruses in the soil under our feet.
Wow.
And so we are in this literal stew of genomic information that's interacting with our biology. And in fact, our biology was designed by it, which is really humbling. And again, it's not the microscope that's giving us the clue. It's the genomics, looking at the patterns of life and how it emerged from the fabric of the planet. And we see now in the human genomics that we now have categorized at least 50% of the human genes as having come directly from an unedited insert from a virus. At least 10% of the human genome is inserted by retroviruses like HIV. And so this is starting to give us a sense of we have vilified the very thing that has built human life on the planet. And if we continue to adopt a scientific attitude of that warfare mentality on the very thing that built us, we will create our own demise. And I believe that's what we're engineering today in our response to a pandemic, in our response to an infection in an ICU. We have this warfare-like mentality that we need to kill everything around us. The risk is that we isolate ourselves. And the second law of thermodynamics is one of the most fascinating laws in science. And it's been proven at the scale of a galaxy, it's been proven at the scale of a single cell, even at the scale of a single atom. And the second law of thermodynamics teaches us that whenever you put a system of large or small capacity or dimension into isolation, its entropy increases, which is its state of chaos increases. What we're doing as a human species right now is so sterilizing ourselves from the environment in which we were born, so sterilizing ourselves from nature in which we developed, that we become an isolated system. And in that, we become extremely chaotic. And we express massive amounts of dysfunction and disease. Autoimmune disease, cancer, major depression, acne. It doesn't matter what you think of. These are all symptomatic of being isolated from an ecosystem that we now understand to be a continuum of life on the planet.
So I'm trying to put this in perspective. We're trying to sterilize everything around us, right? Everywhere you go, hand sanitizer, right? You put on your mask and create this sterility around you, and that is considered protection. But in the model, I think in the way that you're laying it out, you're saying, wait a minute, there's an extraordinary amount of these things around, and when we start to do this sort of sterile practice and try to attack specific microbes, you're kind of throwing off a balance and creating an imbalance in the way that we have to harmonize with the environment. Is that accurate?
Yeah, this sterile bubble, interestingly, as we produce it, leads to a breakdown in self-identity, which is very difficult to believe because it feels like warfare and strong borders of a nation and walls against intruders or immigrants. That seems like strong self-identity. But what we learn at the biologic level is that as we remove the microbiome from the gut, for example, we lose the boundary of the gut membrane. And so the further you sterilize and lose the wireless communication network that's produced by that microbiome or that diverse ecosystem, without that communication of the biodiversity, you can no longer know what's going on at the cellular level and what your response should be. And so we see that the gut suddenly stops making the proteins that are the Velcro that hold it into this profound boundary event that should say this is human beyond this space. And instead we see this erosion of that human identity at the biologic level as we denude ourselves of the microbiome. And so we have this extraordinary realization, I think, as a species, or we have the opportunity for this realization, in that it is only in the participation of a biodiverse ecosystem of the planet that we will reach our full biologic potential. And this reverses or challenges, certainly 150 years, but really a couple hundred years of germ theory, that we thought we had to kill all of those things. We thought they were attacking us. And again, genomics has told a very weird story, that the microbiome is not this organic soil limited to our gut. We now know that the liver, the kidneys, the bladder, the prostate, the breast, and now even the human brain is recognized to have vital microbiome within it in the healthy state. And when we find ourselves in a state of biologic stress, the first response system, perhaps the most intelligent response system to move us toward repair and regeneration, is actually that microbiome within the human system. And so there's this extraordinary new challenge to realize that a healthy immune system, as we've termed it, is not even supposed to kill the organisms within our body. It's supposed to understand and realize a balanced state or a homeostasis with the biodiversity within us and around us. It's never attacked anything. It's only been there to establish balance.
With vaccines, as we look at them, we have historically we can observe there's been many viral plagues, if you will, or pandemics that have come around, have killed some people, and then have gone away. There's never a vaccine for them. It seems like a part of the natural cycle of life to a large extent. And sometimes maybe they're associated with environmental changes, maybe how we moved from more rural settings to concentrated cities, that kind of a thing. So does that play into your model or how you see things?
I think it plays both into this new understanding that's emerging that everything is a continuum of biodiversity, but it's also the very thing that vilified the viruses to begin with, in that we saw these pandemics, or we see flu season every year, or whatever it is, and we assume that the virus is doing this because we can create this scientific correlation that this person is presenting with upper respiratory symptoms of mucus production and fever, and we can measure a virus in their bloodstream at that moment. And so we've made this correlation in time, and we've accused the virus as being the causative agent there. But if we back out and look at human history for sure, and when these things show up, it's starting to become very obvious that the symptoms that we're developing around these viral experiences are the symptoms of toxicity of the environment that we are creating around ourselves as a people, and therefore the virus is uncovering this toxicity. And nowhere more true is that than coronavirus as we've seen it. Coronavirus, as it's moved around the world, it has demonstrated to us where is the highest toxicity in places. It, of course, was first defined in Hubei province in China. Whether it comes from Hubei province originally or not will take us some time to tease out. But what we can say with confidence is that Hubei is the most toxic environment that we've produced on the planet in regard to its soil and water and air systems. And so when we see biologic stress at the level of soil, water, and air, we know we're going to start forcing a large extinction event to start to emerge. And when we force extinction, when we force such high biologic stress that the species of any form, microbiology or otherwise, they start to exude viral information looking for an adaptation event. And so when a bacteria are stressed in the intense herbicide environment of Hubei province that sprays more Roundup than any other province in China, and more intense antibiotic usage in pork industry than anywhere else in the world. And so we have this massive antimicrobial warfare going on in Hubei, and we're creating this huge stressor. And so there's going to be this exudation of viral genomics into that environment that's actually looking for an adaptive possibility. And we have this opportunity to start to see now around the world as we start to follow this pandemic that it crops up in places that don't make sense, really. Why did it go from Hubei province to northern Italy? And then many months later, you see Germany and other places. It's not geographically making sense in its distribution until you look at the toxicity of soil and water and air systems. And so it turns out that Memphis and Louisiana, New York, northern Italy, all that which cropped up within weeks of each other, are the areas of highest spraying of antibiotics and herbicides and pesticides, and the highest air particulate pollution of something that we call PM 2.5, particulate matter 2.5. And so we've created a big stressor, demanded viral production. The virus then can bind to PM2.5 in abnormal clumps, so that, not as nature designed it, we end up with an over concentration of this genomic update or expression, and then that's tagged to a receptor through coronavirus to our lung. And so we're creating this, smart bomb, where in fact the virus was a very benign update originally. Coronavirus obviously causes the common cold. It cycles through in a more virulent fashion every 8 to 10 years. We saw it in SARS, we saw it in MERS, 2002, 2012, 2020. And so we see these patterns of coronavirus happening around the world. And of course, we can go all the way back to like 740 years ago to see the first of these coronavirus-like pandemics. And so it's been in the human experience for a very long time. And it seems to be intelligent in the way in which it expresses the genomic update. And so should not be surprising that we see the highest concentration of genetic expression of this compound where we have the highest degree of toxicity or pressure on biology to change. Toxicity creates the need for adaptation and regeneration. Viruses are the mechanism by which we do that. And so when we see a virus crop up, we need to alter our perspective to not damn the virus, but recognize the viruses are a friendly red flag saying, you got a problem over here. You've got a high degree of toxicity that's happening here. You need to shift this terrain. You need to change the environment here. And that's exactly what we saw with this pandemic, is in Hubei province that has the highest PM2.5 air pollution in the world. As soon as this pandemic lockdown happened, the pollution started to drop. No cars, energy production goes down. As soon as it dropped below 40 particular points of carbon per cubic meter, we suddenly saw the death from coronavirus go away in that province. And so as we clean up the environment from a change in human behavior, we think, we beat the virus, or we somehow have herd immunity to the virus. The virus actually doesn't even interact with our immune system like a bacteria or a fungi does. We're not waiting to win the war against the virus. It's not how it works. That virus is always going to be present in the environment now. We're never going to eradicate coronavirus. It's going to be around forever. And it's going to be different variations all the time. We're already creating variations of what we call COVID-19. That already has multiple genomic variations in it. Even within the United States, in those first few weeks, we're already at 3 different strains of that genomic sequence. And so to say that there's one thing that's attacking us is super inaccurate. There's one stressor that we're creating that's expressing itself in disease. And the virus is, in many ways, the innocent bystander that happens to be trafficking the pollution into our bloodstream through its smart delivery system, not knowing or not being able to modify the fact that it's carrying air pollution into our bloodstream. PM2.5 binds the virus, and it also binds things like cyanide and the other common toxins that we produce in the energy industry. And so we're pulling the toxicity of our air pollution right into the bloodstream by the action of a smart virus that never intended any harm. It was an adaptation to biology in probably the most important organ systems that are being challenged by that very toxicity. In areas of high PM2.5 and cyanide, your vascular tree, and in fact the hemoglobin that carries oxygen, is the place that we need the most adaptation. And it's pretty amazing that more than 50% of people that have been exposed to coronavirus don't have any symptoms. They create some antibodies, they find balance with it, they express this genomics within their body, and they get that update in the lung, in the vascular system, and in the red blood cell, importantly in the hemoglobin. There's an adaptation event that happened in those people. And it's not going to surprise me if in 10 years we find out that the people that were exposed to coronavirus, asymptomatic or otherwise, are suddenly resilient against some other, foul play of humankind, and they're more resilient to lung cancer, or more resilient to COPD or chronic bronchitis or autoimmune disease, because they successfully got this genomic update in the very tissues that we would expect dysfunction to happen.
So this is fascinating, because now it really does kind of turn the current perspective on it, right around 180 degrees, where you're looking and saying that that literally it's delivering a biological upgrade. And in some people, especially in compromised circumstances, as you cited, it's the environment that it's functioning in that's carrying these things in, because that's what it's designed to do. Not meant to hurt us, but meant to be able to bring these things in and allow adaptation to occur. So how do we create, or do you see it's emerging, that there's a new model of germ theory, if you will, as compared to germs are bad and we have to attack and go to war with them. And winning the war means creating vaccines, which can be overwhelming and damaging to our immune systems as compared to supportive. That there's a different model that says, we need to understand this, and now here's how we can operate. And the vulnerabilities, as you cited, are in certain areas where man-made environments have allowed for this coronavirus pandemic to occur.
Yeah, we have to come to terms with the fact that we keep finding at the foundation of dysfunction, pathophysiology, or disease, we see a perturbation in the microbial diversity first. And so as we create an unbalanced or damaged ecosystem, then we can expect and count on dysfunction within the human biology. And this emerged from the genomics of the gut microbiome in the 2005-2010 era, when we started to first do genomic sequencing of all the bacteria in your gut. And then we were starting to correlate that with human disease, and to find out that if you were missing these bacteria, you start to get breast cancer. If you miss these bacteria, you'll get colon cancer. If you miss these bacteria, you get lung cancer. And so this extraordinary discovery of how is it possible that these gut flora, these bacteria that have nothing seemingly to do with the lung, are predicting in their absence the occurrence of lung cancer. And when I was developing chemotherapy back in the day, that was so antithetical to our model of cancer that we would literally sit around our lab meetings and laugh about these hippie journal articles that were coming out of UCSD and UCSF. And I'm in this pseudo ivory tower of the University of Virginia, a wannabe kind of Ivy League environment of like, well, we're far above that belief that bacteria, because we know that cancer is actually this accumulation of genetic injury in a human cell, and we have this very human-centric belief system about what cancer is, and the bacteria simply didn't fit in. And so we'd laugh that off, but pretty soon it got so ridiculous in the amount of data that's coming out of the genomics, it passed the gut. So by 2014, studies were starting to be published showing that the microbiome within the human breast could be demonstrated to have a dominant species of Sphingomonas in the healthy human breast. And as you start to stress that environment and create an acidic tissue environment from chronic emotional stress, chronic environmental stressors, food stress, all of these things, the Sphingomonas cannot thrive there. And so you see Sphingomonas step out of the microbiome, and you see a Mycobacterium radiotolerans step in as the dominant species. And so then there was this brief belief that, Mycobacterium must be causing breast cancer.
As I say, does it get confused because it's coincident that, this must be the cause because it happens to be there as compared to the cure?
It was exciting. We were like, maybe it's like H. pylori causing, ulcers in the stomach. Maybe the breast cancer, because it was every breast cancer they studied had this bacterium present, had other bacteria too. So it's interesting that both the diseased breast and the healthy breast have an ecosystem, not just the Sphinx, but dominant Sphingomonas. And the study that was really telling was one that studied the bilateral breasts in the same women. So a breast with breast cancer in it, and then the contralateral breast with no cancer in it, had different gardens, had different organic material, different microbiome in there. And so they were set out to show that the methylbacterium was causing cancer. And so to do that, they wanted to show that the more genomic DNA sequencing of methylbacterium, the more aggressive that cancer got. And in fact, they showed the exact opposite. The more sterile that tumor got, the faster that woman died of metastatic cancer.
Wow.
And so it was this startling realization that the shift in the microbiome was the first step towards damage control. And it was trying to mobilize resources within a breast that can no longer support Sphingomonas because it's an aerobic bacterium. Methylbacterium is an anaerobe, and it can deal with a lack of oxygen, and it can still produce energy. And so in tissue that's starting to lose its tissue potential of energetic repair and mechanisms of regeneration, methylbacterium steps in. If we then hammer that bacteria with antibiotics and chemotherapy and everything else and eradicate that bacterium, we risk the reality of a metastatic event and death from cancer. And so in the same way that we're having to come to terms with the fact that the microbiome is smarter than human biology itself, the bacteria know how to adapt to human cellular injury better than the human cell seems to. The human cell can't recover without the reemergence of that gut microbiome and tissue microbiome that needs to be present. And we can't stop that at the viruses. We can't then say, oh yeah, well that's the bacteria that are acting like that, but the viruses are all bad. And that's definitely the attitude that you'll find in scientists. Well, viruses are all bad. They're these Trojan horses that take over our genetic apparatus and produce a bunch of viruses to attack other cells around and all of that. That's ignoring the last 40 years of genetic data as to just how regulated the transcription of RNA and DNA is in our cells. It's the most regulated event that happens in the body, is to determine which RNA strand should make a protein, when should DNA make a specific RNA. These are very highly regulated steps with thousands of feedback loops and communication systems attached to it to make sure it never happens in an unregulated and detrimental form. Viruses can't come in and co-opt that whole system. It's impossible. There's too many checks and balances in the system. A virus has to come in and be welcomed into the genomic sequencing and the production of proteins. And the immune system, as we understand it, with antibodies attacking these viruses and everything else, is another fallacy. We don't form a antibody to the virus itself. We form an antibody only after the RNA has decided to replicate and put this genomic information into larger circulation into that body. And so a single cell that starts to manufacture that RNA resulting protein and sticking these new proteins into our bloodstream that can then go and transmit to another cell and get that whole genomic update to all of the locations it's supposed to be. Remember, this coronavirus isn't going to hit all of the tissues of the body. It's only going to hit the ACE2 receptor and where it's needed. And so then the cell, while making that protein, will deposit little protein pieces on its surface for the surveillance of the immune system to say, is this stuff that we need or not? And if the immune system decides that there's a new protein being made by this cell that's not wanted in the system, it will bring in a killer T cell and a macrophage and clean that cell up so that it stops producing that protein or the subsequent viruses. But it's way downstream that an antibody ever gets involved. And this is, starting to demonstrate that we need to expand.
Explain why that's significant, that it's way downstream that an antibody gets involved, because that's the whole fundamental basis for why they want to create vaccines to create antibodies, but they're missing the big picture.
Yeah, unfortunately, we don't come into balance with viruses through antibodies. And that's, of course, very fortunate, because we'd die if we were dependent on the antibody, because the antibody can't prevent the infection from happening. It can only bring us into balance after the genomic update is in process. And in fact, it can't eliminate the virus itself, because it can only control the amount of cells that are expressing that thing over any period of time. And like I said, 50% of the genome of the human was stacked by these viral inserts. If we had the intention of clearing out all of that genetic information, mammals could have never occurred. And these are critical genes. If we look at human life, it turns out that the placenta, the formation of a placenta within the womb of a woman, depends on an RNA virus that updated our genome millions of years back. We wouldn't have had the first birth of a mammal without an RNA sequence integrating itself into that biology. Perhaps just
so you're saying mammals became possible because of viruses?
Entirely. We would not have had the first mammalian birth without it.
Wow.
And furthermore, we would have never repaired. So I don't think we would have lived past a year of life if we didn't have another RNA sequence that actually is the way in which a stem cell is enabled. So the stem cell, which is this pluripotent mechanism of regeneration within our body, can replace any cell within the body if too damaged to replicate. That stem cell has this very specific RNA gene that allows it to move on the genetic sequence of the whole human DNA across bumper blocks. And so there's these hard bumper blocks between genes typically that make sure that this cell is just going to be a liver cell, and this cell is just going to be a lung cell.
And that's how you get differentiation. Yeah.
To get a stem cell to do any of those, you need a new RNA sequence from a retrovirus like HIV that got integrated into the mammalian genome to allow a stem cell to emerge that can do any of the above. And so these critical mechanisms of human life are showing us that the immune system's intention was never to eradicate the genomic updates. It was always to bring us into balance with that genetic information. And so how does that occur if it doesn't happen by antibodies? And this has been discovered largely through the study of HIV and hepatitis C. These chronic infections that are present in the body over decades and decades have allowed us to come to understand that the balance or homeostasis that we achieve with HIV is actually done by these little scissors that are produced inside the cytoplasm of the cell. And so there's these very specific enzymes that target RNA or DNA of viruses specifically and clip up the DNA before it can go into a replication state. And those enzymes are busy. And so 10 to the 31 viruses in the air around us, I can guarantee you over this short conversation, I've probably chopped trillions of cells within my body that are chopped up DNA sequences within trillions of times over just an hour of time, because there's so much being absorbed all the time. So I have to be in this constant editing mission. And so we edit the genomic information before it ever goes into a sequencer production. That's why the vast majority of people out there that will live in a family that somebody has coronavirus in actually won't get the condition at all. And then half that do get the condition will be asymptomatic or mildly symptomatic. It's because they're editing all the time. They're not waiting for some antibody to form weeks later to eradicate this virus. They're able to come into this homeostasis balance through this enzymatic work.
Isn't Dr. Zach Bush just a beautiful soul? So excited that I could share this with you. I hope that you'll take this message and share it with others. Let people know about Vaccines Revealed. Let them see the luminaries that we're interviewing. I appreciate you tuning in. And part 2 is going to be coming up for Dr. Zach Bush, so make sure you're there to see that also. Thanks for being here.